Welcome back to the deep dive. Today, we're getting right into the nitty-gritty, the real molecular underpinnings of forensic biology. Specifically, we're tackling serology concepts.
Yeah, this is foundational stuff. Serology, it's often the first step in the lab for identifying biological fluids. It all comes down to these antigen-antibody reactions.
Exactly. And our goal today is basically to give you a kind of conceptual cheat sheet. We want to clarify those key chemical players, the reagents, the reactions.
So that when you hear about specific forensic assays later, you'll actually understand why they work the way they do. It's about the rules of engagement at the molecular level.
Okay, let's start with the targets then. We often hear two terms, immunogen and antigen. They sound similar. What's the key difference for us?
Right. It's subtle but important. Think of it like this. An immunogen is something foreign, usually quite large, like a protein or a complex sugar molecule, that when it gets into the body, it can actually trigger an immune response. It makes the body produce antibodies. Think blood group antigens like A, B, O. Those are immunogens.
Okay, so immunogen means it causes the reaction. What about antigen then?
An antigen is any foreign substance that can react with an antibody. So here's the key. All immunogens are antigens because if they cause antibody production, those antibodies can definitely react with them. But, and this is the crucial part, not all antigens are immunogenic. Some things can be recognized by an antibody, but they can't actually kickstart the whole antibody production process on their own.
Ah, okay. And that leads perfectly into something really cool in forensics. The hapten. Explain those.
Yes, haptens are the perfect example. These are usually small chemical molecules, think drugs like cocaine or amphetamines. An antibody can bind to a hapten, so it's antigenic, but the hapten molecule itself is just too small, too simple to trigger the immune system to make antibodies against it.
So how do we make tests for them then, if they don't cause antibody production?
That's the clever part. In the lab, scientists take the hapten and chemically link it, sort of glue it, onto a big carrier protein. This whole hapten carrier complex is big enough to be an immunogen. So you inject that into an animal, the animal makes antibodies, and crucially, some of those antibodies will be specific just for the hapten part. Then you can harvest those antibodies and use them in a test to detect the free, unattached hapten, the drug itself, in a forensic sample. That's basically the foundation of many drug immunoassays.
Wow, okay, so you're essentially tricking the immune system to get the tool you need. Makes sense. Now, when an antibody binds, what part is it actually grabbing onto?
It binds to a specific little patch on the antigen called the epitope, or sometimes determinant site. It's the unique shape or chemical structure the antibody recognizes. And since most immunogens are large and complex, they usually have multiple different epitopes on their surface. That's why we call them multivalent.
Got it. Multiple binding spots. Okay, let's switch gears to the tools themselves. The antibodies, or immunoglobulins.
Yeah.
They're different classes, but why is IgG considered the main workhorse in forensics?
Well, there are a few reasons. IgG is the most abundant type in our blood serum, which helps. But more importantly, IgG is the star of the secondary immune response. That means it's produced in large amounts during a later, more mature immune reaction and sticks around for a long time. It has a longer half-life.
Which is good for forensic samples that might be old or degraded.
Exactly. On aged or dried stains, your chances of finding intact functional IgG are generally better than, say, finding the IgM that pops up really early in an infection and then fades.
Okay, and structurally, we always see that Y-shaped picture. Can you describe that?
Sure, so IgG looks like a Y. It's made of 4 protein chains, 2 identical heavy chains that form the stem and part of the arms, and 2 identical light chains on the arms. They're all linked together by disulfide bonds. The really important feature is that it's bivalent. Each tip of the Y arms has an identical antigen binding site. So one IgG molecule can potentially grab onto 2 antigens.
Right, 2 hands to grab with. And that bivalency, that's going to be critical later when we talk about clumping and precipitation, I assume.
Absolutely. It's essential for forming those visible networks.
Okay, so labs need these antibodies as reagents. They can use polyclonal or monoclonal. What's the difference in practice?
Big difference. Polyclonal antibodies are kind of the old school method. You immunize an animal, its immune system reacts to all the different epitopes on that immunogen, and then you collect its blood serum, which contains a whole mixture of different antibodies produced by different B cells, all targeted against that same immunogen, but binding to slightly different spots.
So it's like a cocktail of antibodies.
Pretty much. They tend to be robust, good for recognizing the antigen, even if it's a bit damaged. But because it's a mix, there can be variations from batch to batch. So you constantly have to validate and potentially adjust your test parameters.
Okay, versus monoclonal antibodies.
Monoclonals are the high-precision tools. They're made using a lab technique involving hybridoma cells, basically fusing an antibody-producing cell with an immortal cancer cell. The result is a cell line that pumps out huge amounts of one specific type of antibody, an antibody that recognizes only a single epitope.
So super specific, consistent, available forever. Sounds perfect. What's the catch?
The catch is related to that bivalency we mentioned. Because a monoclonal antibody only recognizes one specific epitope, if you mix it with a soluble antigen, it often struggles to form those large cross-linked lattices needed for a visible precipitation reaction. It can bind, sure, but it might just bind one antigen molecule, or maybe two, but doesn't easily build that big network that falls out of solution. Polyclonals, with their mix targeting multiple epitopes, are much better at that. Ah, okay. So the choice of antibody type directly impacts what kind of test you can even run. That's a huge practical point.
Definitely. And one last region type to mention quickly, antiglobulins. These are just antibodies against antibody.
Antibodies targeting antibodies, why would you need those?
Well, often in a test, your primary antibody binds the target, but that binding isn't visible. So you add a secondary antibody, an antiglobulin, that recognizes a common part of the first antibody, like its stem or Fc region. And that secondary antibody might have a label attached, like an enzyme or a fluorescent tag, or it might help with cross-linking. It's a way to detect the detector, essentially. Very common tool.
Okay, makes sense. So we have the players, the targets, and the tools. Now let's talk about how strongly they interact. You mentioned the binding is non-covalent, rapid, reversible. How do we measure the strength? We use two main terms here, affinity and avidity. Affinity refers to the strength of the interaction between one single binding site on the antibody and one single epitope on the antigen. Think of it like the quality of the lock and key fit. High affinity means a very tight, specific interaction for that one site.
And that specificity is important, right? To avoid accidentally binding the wrong thing.
Exactly. High affinity helps minimize cross-reactivity, where an antibody might weakly bind to an antigen that looks similar but isn't the actual target. Lower affinity binding is usually involved there.
Okay, so affinity is the single site strength. What's avidity?
Avidity is the overall binding strength between the antibody and the antigen. Because IgG is bivalent, two sites, and antigens are often multivalent, many epitopes, avidity takes into account all the binding interactions happening simultaneously. It's like Milkrow. One hook and loop connection, affinity might be weak, but thousands of them working together, avidity make it really strong and stable.
So avidity is the combined strength, the synergy.
Precisely. And in the lab, high avidity is really important because it means the antibody-antigen complex will stay together through washing steps and other manipulations in the assay. It gives the test stability.
Got it. Affinity for specificity, avidity for stability. Okay, now let's get to the results. How do we actually see that binding happen? We've got primary and secondary reactions.
Right. We can mostly skip tertiary reactions. Those are biological effects in the body, like inflammation. We don't really measure those directly in forensic in vitro tests.
So primary reactions.
The primary reaction is just that initial binding event. One antibody site meets one epitope. It's fast, it's reversible, it forms the basic ag-ab complex. We quantify this equilibrium with something called the affinity constant, kappa. Higher kappa means stronger binding. And these primary reactions themselves form the basis of our most sensitive techniques, things like ELISA or radioimmunoassays, where we detect that binding indirectly.
But they aren't usually visible on their own.
Generally not, no. For visible results, we rely on secondary reactions. These are usually a bit less sensitive, but often easier and quicker to perform, especially for initial screening. The main ones are precipitation and agglutination.
Okay, let's break those down. Precipitation first.
Precipitation happens when you mix a soluble antigen with its antibody, which we sometimes call a precipitin. If the conditions are right, specifically the ratio of antigen to antibody, the bivalent antibodies cross-link the multivalent antigens, forming huge molecular networks. These networks become so large they're no longer soluble, and they fall out of solution as a visible solid, a precipitate.
And getting that ratio right is governed by the famous precipitin curve, isn't it? Tell us about those zones.
Yes, the precipitin curve. If you plot the amount of precipitate formed against increasing amounts of antigen added to a fixed amount of antibody, you get this characteristic curve with 3 zones. In the middle, you have the zone of equivalence. That's the sweet spot. The ratio of antigen to antibody is just right for maximum cross-linking and lattice formation. You get the most precipitate here. That's what assays aim for.
But what happens if the ratio is off?
big problems. If you have way too much antibody compared to antigen, that's the prozone or zone of antibody excess. Each antigen molecule gets instantly coated with antibodies, but there aren't enough antigens to bridge between them. No effective cross-linking, so no precipitate
So
forms.
too much antibody can lead to a false negative.
Absolutely. And the same thing happens at the other end. If you have way too much antigen compared to the antibody, that's the post-zone or zone of antigen excess. Now, every antibody binding site gets saturated with a separate antigen molecule. Again, no effective bridging or cross-linking between antibody molecules can happen. Result, no precipitate, another potential false negative.
Wow, so you have to hit that zone of equivalence,
Hmm.
which means sample dilution before testing must be incredibly important.
Critically important. If you get an unexpected negative, especially with a concentrated sample, you should always suspect prozone or post-zone effects and retest with dilutions.
Okay, that makes perfect sense.
Yeah.
Now, the other main secondary reaction, agglutination. How is that different?
Agglutination is very similar conceptually, but the key difference is that the antigen isn't soluble. It's located on the surface of something larger, like a cell, maybe a red blood cell, or tiny synthetic particles like latex beads that we coat with antigen. When you add the antibody, instead of forming a precipitate, the antibodies bridge between these cells or particles, causing them to clump together into visible aggregates. If it involves red blood cells, we call it hemagglutination.
Like blood typing tests.
Exactly like blood typing. That visible clumping is agglutination.
And is there a process to how that clumping forms?
Yeah, it's generally seen as a two-step process. First, there's the rapid initial binding of the antibody to the antigens on the cell surface, just like the primary reaction. Then comes the slower second step, lattice formation. This is where the antibody, already bound to one cell, manages to reach out and grab onto an antigen on a different cell, physically bridging them together and building up that clumped network.
And does the type of antibody matter here?
Oh, hugely. Remember we said IgG is bivalent, but relatively small and compact. While it's great in many ways, its size can sometimes be a disadvantage in agglutination. It might bind perfectly well to an antigen on one cell, but it might physically struggle to span the distance needed to reach and bind to a second cell, especially considering repulsive forces between cells. We call antibodies that bind but fail to cause visible agglutination incomplete antibodies.
So IgG can sometimes be an incomplete antibody, even though it binds.
Yes. It binds in the first step, but fails at the second lattice formation step. This is where the other major antibody class, IgM, really shines. IgM is this huge pentameric molecule, like 5 IgGs joined together, with 10 potential binding sites. Its sheer size and reach make it incredibly efficient at bridging between cells and causing rapid, strong agglutination. It's much less likely to be incomplete in this context.
Fascinating. So the physical structure of the antibody directly impacts its ability to give a visible result in these different kinds of tests.
Absolutely. It's all interconnected. The nature of the antigen, the type of antibody, their relative concentrations, even the physical size and shape.
All right, let's recap then. This has been a really helpful breakdown. We started by sorting out immunogen versus antigen and saw how that distinction lets us use haptens for things like drug testing.
Right. We looked at the IgG antibody structure, its bivalency, and contrasted the mixed bag of polyclonal antibodies with the highly specific but sometimes limited monoclonal antibodies.
Then we clarified affinity, that single-site binding strength driving specificity versus avidity, the overall stable grip crucial for lab tests.
And critically, we explored how precipitation and agglutination work, emphasizing the absolute need to be in the zone of equivalence to avoid false negatives from prozone or post-zone effects.
And finally, understanding that even a great antibody like IgG can be incomplete in agglutination tests simply due to its size, failing to form that visible lattice.
Exactly. These aren't just definitions, they are the working principles. Understanding why you might get a prozone effect or why a monoclonal might not work in a precipitation assay is vital for accurate forensic interpretation.
It really puts the practical lab work into context. Which leads us to our final thought for you, the listener. We talked about that limitation of monoclonal antibodies, fantastic specificity, but poor at forming precipitates because they only bind one epitope. So here's the challenge. If you wanted to use a highly specific monoclonal antibody in a test that required a visible precipitate or aggregate, how might you creatively design the assay to overcome that single epitope binding limitation? Think about how you could artificially force those complexes to link up.
Some clever ways around that exist. Something to ponder. Thanks for diving deep into these foundations with us.
Yes, thank you. Catch you on the next deep dive.